morning, ladies and gentlemen, and welcome to the third day the International Course Bioinformatics and Neuroscience 2016. To start this morning of conferences, it's my entire privilege to introduce you to Dr. Leonardo Mariño from the National Center for Biotechnology Information, the United States. He holds a bachelor's degree in microbiology from the University of Los Angeles and also holds a PhD in biochemistry from Texas A&M University. He has written extensively in situ identification of evolution gene regulatory elements, molecular and cellular interactions, controlling gene expressions in eukarya. He also has experience in the database design for biological data. So, given nothing more to say, let's welcome him with an applause, please.
Thank you very much for the introduction, and I would like to thank the organizers for inviting me here to Janet and also Nelson here. I'd like to by telling you that I'm happy to be back. I'm from Colombia, and I'm really happy to see people from different countries represented here. So let's start by saying that I'm gonna be talking about cloud computing resources and for bioinformatics and next-gen sequence analysis. So I come from the NCBI. The NCBI is the National Center for Biotechnology Information. We're located in Bethesda, Maryland. It's a suburb of Washington, D.C. We were created in 1988 as an act of Congress, and we are underneath the National Library of Medicine. So our mission is to establish public databases. The public databases were hosted at Los Alamos laboratory in right before 1998. We also do research in computational biology. We develop software tools for sequence analysis, and we also disseminate this biomedical information freely to everybody. Now, this announcement was done probably a couple of years now. We have now the 1,000 genome is here. And Illumina is the company that is probably the market leader right now, not only for research applications, but also for clinical applications. Now, the genome has changed tremendously since the human genome was declared complete back in 2003. So we have that right now we have more complex genome structures. We have chromosomes, we have organelles, we have plasmids, we have genome sequencing that has enabled this revolution. We have more complex genome assemblies. We used to have complete chromosomes. Nowadays we have scaffolds, we have context. That doesn't mean that we're not able to get information out of them, but we need to be aware that complete genomes are not longer the norm. So that has to we have also genome scale projects. These projects include transcriptomes, exomes, epigenomics, proteomics, and other omics type of projects that enable us to study different species, not only model organisms, but do genomic analysis or other type of omic analysis in, for organisms that are not model. Now, we also have multi-isolate sequencing. So we have, for example, the 1,000 Genome Project. We have learned a lot about population genomics in the last decade. And we also have metagenomes. And now I think that this is useful for drug development. Let me also start by showing this graph. This is the cost of an entire human genome starting in 2001. The price tag for that was $100 million. Okay, look at the scale. The scale in the Y is in log scale. So every time that you see a linear decrease is actually an exponential decrease. So what we have here is an exponential decrease from 2001 up until, let's say, 2006, 2007. No, sorry, 2007, 2008, when next-gen sequencing became very popular. So we have that the whole genome has dropped in price significantly. And the last number, because the NHGRI really wants to track this. They want to make sure their money is well invested, so they're tracking how much genome sequencing will cost. This was very close to the $1,000 in 2015. So now we have a big challenge. And that challenge is, how do we go from this data to biological knowledge? So now we have a bioinformatics bottleneck. And one of the, or few of the challenges are in different areas. First, in methods, we have, how do we analyze the data using procedures for different various data types? We also have challenges in terms of infrastructure, and I will talk about that more. It's, where do I process my data? Large-scale computing, accessibility, installing, and also maintaining software and hardware as well. And also another challenge is in standards. How do I ensure that my results are useful? We need to develop common and share standards to be able to share data and analyze this data as well. So back in 2010, Lincoln Stein proposed, or he made a case for cloud computing. And that's something that we're starting to see today. And so the model that we have, we still have this, is we have sequencing labs from all over the world providing raw data to the databases. So the primary repositories, let's say NCBI or the EBI or the DDBJ in Japan, right? Now, those sequence archives are the primary archivers. Now, we have also some value-added integrators like Ensemble or UCSC. So in this case, they will take these primary repositories and add another layer or multiple layers of information to complement this type of data. So as you can imagine, there is a lot of data being not only generated, but also being transferred constantly, very frequently between the sequencing labs, the sequence archives, and those value-added integrators. And we also have the end users, like yourself, for example. You want to go to the primary databases or to these value-added integrators and get some queries. We also have power users, which may be large sequencing centers that want to keep their data in sync so they can analyze and annotate genomes. So what happened was, and this is also another figure that shows log scales. The left axis corresponds to disk storage in megabytes per dollar. So as you can see in this particular figure, this is also in log scale. We have time in this scale here from the 90s up to 2012. And we see that before next-gen became very popular, the growth was at this particular pace. Now, the hard disk storage was growing way faster up until when we started seeing next-gen becoming very popular. So right at this stage, which is, let's say, 2009, 2010, we started to seeing that next-gen is, the output from next-gen is outpacing the way technology is being generated for disk storage. Now, that may not be a problem for you guys, but it's definitely a problem for NCBI. So one of the things that we're trying to do is to develop ways to compact the data, to compress the data, and store it in a better way. So the case for cloud computing, and this is we're starting to see this right now, fortunately, is we have the sequencing labs that submit data to the sequence archives, and then the primary sequence archives share a cloud environment with these virtual clusters, the value-added integrators, and then the casual user or the power user will access these cloud resources to be able to access the data more efficiently. So this is a more sustainable model, and I'm really glad that we're having that right now. So I'm gonna change a little bit about some of the cases that we have done at NCBI. The first one is bacterial gene annotation. So we have a pipeline for genome annotation at NCBI in terms of for prokaryotes. So let's say that you have 100 sequences that you have assembled for microorganisms and that you want to get annotations for. You can submit that to us, and we can generate, at this time we have about 400 assemblies per day. We want to replace that data flow to create also RefSeq assemblies. So one of the, let's say, challenges that we had before RefSeq was established was that the primary submitter owns the accessions. So those accessions can only be modified by the author. And so let's say a genome was sequenced, let's say Mycobacterium tuberculosis was sequenced 15 years ago or 10 years ago. That accession will not be modified by the author. The author is usually interested in publishing the paper and move on to the next organism. So what we have with RefSeq is that the RefSeq assembles are being revised every time we get better technology, better annotation technologies to keep the latest and the most up-to-date annotations in the databases. So we are also performing annotation via mapping from closed assemblies, and this is also useful because, for example, recently we had the discovery of CRISPR-Cas system. The CRISPR-Cas systems were not annotated. They're very useful, but were not annotated 10 years ago, for example. So this is all now being revised. So let me talk about the particular process that is done. So we have an assembly that's provided by the submitters. We identify mobile elements. We have non-coding elements like ribosomal RNA, tRNA, non-coding RNA. We have a protein placement. With all this, we resolve conflicts. We use also GeneMark, which is an ab initio gene prediction tool. We expand that evidence. We use also GeneMark to then assign names and prepare reports. This is in the case where we don't have a closed assembly. Now, in the case of having a reference assembly that is very close, that could be useful to annotate this particular genome, and fortunately this happens more and more, we can use that to align the assemblies for the reference and the de novo assembly that was done. Then we get this, we project this protein annotation, we resolve the conflicts, we go like that. But of course, a lot depends on the quality of the reference set, right? We may optionally choose to project non-coding annotations as well. So one of the things that we're doing is this pathogen analysis, and this is important because we're collaborating with labs in the US, and not only in the US but also all over the world, to do a rapid identification of species and strain from next-gen sequencing data. So here we have a rapid assembly and annotation of bacterial short-read sequences, rapid identification of key characteristics separating outbreak strains from background samples, and we have this particular initiative, which is the genome tracker. And this is a collaboration between state, local agencies, federal, foreign public health agencies, where NCBI is participating in terms of the annotation and also taxonomic placement, genome annotation as well, with FDA and also CDC. And this is a project where you guys can participate. Let's say that you're from Colombia, or you're from Peru, or you're from Mexico, or you're from Egypt. Any place you can participate in this particular program, submit your sequences, and try to identify some of the characteristics that are important for these pathogens of interest. So the ones that we have made a priority are for foodborne illnesses, Salmonella enterica, Listeria monocytogenes, E. coli, the O157, Campylobacter jejuni, also some resistant and virulent hospital-acquired infections like MRSA or Klebsiella pneumoniae, and also some of the difficult to culture organisms like Mycobacterium tuberculosis. And this is a paper that we actually published, I think, a couple of years now, in collaboration with Corpoica. So this is one of the examples of the collaborations that we're establishing with Colombian institutes, and we're also happy to establish this with other people to try to characterize this multi-drug resistant Salmonella entericus in the Colombian poultry food chain. Now, how is the pathogen analysis identification done? So we have a biosample, we have the assembly, and we have a procedure that first do a K-mer classification. So K-mers are small pieces of DNA that can be used to match and predict the taxonomic placement of your sample. So you don't need to know what organism this is. And we're also using this K-mer classification to try to help submitters place their sequencing experiments in the right taxonomic location. So things like this have happened. So you may have a sequencing of a Campylobacter coli that the submitter thinks is Campylobacter jejuni, for example, or vice versa. So we're able to, once this analysis for K-mer classification is done, we may tell the submitter, hey, what you think is this species is not that species, but this other, because we have evidence in the databases that the genomic placement should be different one. So sometimes classifying bacteria can be a little bit tricky, particularly in some species. So we're using this particular technique to help submitters correct or classify their submissions better. Now, the future for the pathogens are actually increased throughput. We currently have 400 samples per day. The goal is to get at least, that's the short-term goal, to get 1,000 samples per day for genome annotation. Add this automation task for signal detection, add automated submission and report back to the submitter. Predicting virulence and drug resistance as well in the reports, extending to more organisms, and also provide public browsing resources so people can actually make use of this. Now, coming back to the specific cloud, we have made BLAST available in the cloud. And now, why are we making this available? BLAST is probably one of the resources that we have at NCBI that is most heavily used. Now, the databases are growing tremendously. So we have a doubling time probably every 11 months. So if we have a database today in 11 months, we have twice the data that we had that same day. Okay, so it's growing really fast. But in addition to that, we get submissions for doing blast searches from labs all over the world. And we are getting to a point where we need to distribute this compute in a different type of setting. And we decided that the cloud is actually a great way to do that. So we have made virtual machines that are available in the Amazon Web Services. We have also in the Google Compute Engine and also the Microsoft Azure clouds. So those are probably 1 or 3 of the most popular cloud environments today. So we have images for all this blast, and also the databases, we keep them on those cloud environments. So you don't need to worry about, am I working with the latest version of the databases or the latest software? Everything is being maintained by us. We provide this service. The only thing that you guys have to pay is for the compute time that you use in these particular resources. So we have a series of documentations, we have user manuals, we have some scripts that can be used for you. We have also a YouTube channel that tells you about how this works. And we also have a poster that we presented in 2014. So basically, the way this works is you are a user, and again, you can be in the middle of Africa or in the middle of China or anywhere in the world. You connect through your computer, and then you provide the sequences that you want to analyze. You upload them to the cloud. The cloud then does the heavy computational processing and the analysis with the latest version of the NCBI blast executables as well as the databases. You don't have to worry about that. And then you get the result back to the user, right? So we maintain this part. The NCBI executables and the blast databases, we transfer them to the cloud environment. So this is actually a very good model because, let's say, a university like La Javeriana, I'm sure that you guys have very good compute facilities and high performance computing facilities. But if you are in a different, a less fortunate university in let's say, a small town, you would be able to use this as opposed to asking the president of the university or someone to try to build and maintain a high-performance computing facility. So what are the options? So again, we have Microsoft Azure, we have Google Compute Engine, we have BioNimbus. This is an academic cloud. We have any cloud app with the Galaxy project and also Amazon Web Services. So this is the way this would look. Probably you can see it better here, but again, this we have manuals, we have YouTube videos, so you can, if you want to play with this, you can actually do it. And this is a screenshot that shows how this works in Amazon Web Services. So I don't know if you heard the story about how Amazon Web Services came to be, but I'm going to be brief about this. So they had to build a large compute infrastructure to be able to cope with the demand that would happen both at Christmas and also on Black Friday. So those are the times of the year where people place more orders at Amazon. So the rest of the year, that huge compute infrastructure was not being used. So someone said, hey, why don't we rent this compute infrastructure to other people? So this is probably one of the commercial clouds that are more mature today. The prices have dropped significantly. So if you want to play with something like this, you can actually start to do that right now. So once you log in, you can select the different pricing depending on how big your data set is. You can also select a reasonable instance. That means how large your machine do you want that to be, what configuration in terms of memory you were going to have, how many processors, and so on. And then you can launch the instance, right? And once that instance is launched, the processing takes place, and then you get back your results. Now, changing gears a little bit, I'm going to talk about some of the databases that are also important clinically. So this is a search. You cannot read it very well, but here it says cardiomyopathy. You can search by symptom, and this is also useful, particularly for the topic of the seminar. So let's say that you want to get information from NCBI on cardiomyopathy. So here we have a database that is important. It's called ClinVar. You get a number of hits in ClinVar. And once you get to ClinVar, you can also get information about cardiomyopathy here. You can also see information about the pathogenic status of that particular change that you're looking at. You also find an evidence track that tells you if there are any other studies that have been done with that particular variant. Is it conflicting? Is it not? Because sometimes that can happen. So you can actually rank that particular variant, and then you can decide, in this case, and I'm sorry, you cannot read this. So you can decide and prioritize your targets of interest. Now, you can also search by variation status. So you can say, let's say, an NM, which is a transcript, and you want to search for a particular variant, on a SNP, a single nucleotide polymorphism right there. You can also get that information directly from the NCBI databases. You can also search by gene. In this case, you can get information about that particular gene of interest, and also narrow down into some of the exons that might be of interest for clinical purposes as well. Here we have a region that you can zoom in, and you can probably, if you're interested in a particular region that happen to be more frequently found in the variation in the clinical databases, you might be able to get more details about the particular variants that happen to be shown in an exon of interest, on a region of interest. Also, the variation viewer, it's also a new piece of software that we have developed, allows you to look into specific changes that might be of clinical interest as well. And we can also have this view where you can see move around the chromosome and see other regions of interest, and pinpoint the region of interest in the chromosome, but also in the transcript or the genomic region that you're looking at. Here you can, if you, I know this might be of interest for a number of people here, so please, if you want to get more information, you can get to this YouTube link and get more information about that. Now, the variation viewer also has a number of filters that you can check or uncheck to be more specific about the type of experiments that you want to bring. You can get them by year, you can, or no, sorry, by ClinVar status, for example. Is it yes or no here? Is in DivSNP or DVBAR? We have the variant type that you want to look at, and so on. Here, you can also, again, this is an example where you can check different boxes. So, for example, in this case, I want to choose only the pathogenic ones, or the ones that are most likely to be pathogenic, and then you get less results than the ones that you get here. So with this, I would like to finish and close. I would like to thank Richa Argawala. She is a researcher at NCBI that does genome assembly. David Landsman, the branch chief of CBB. Christian Camacho is a friend, a Peruvian, that is working on the cloud deployment of BLAST. Pilar Donado, a collaborator from Corpo ICA and the intramural research program of NCBI and NLM for funding. Thank you.
So thank you for your talk. Now we have almost 15 minutes for questions and answers. Does anybody from the forum has a question? No? Okay.
I'll break the ice.
Thanks.
My question has to do with metadata annotation. In the case of genomes for species, that's fairly settled, and NCBI contains one of the best taxonomies that are publicly reachable. As we move into single-cell sequencing,
right.
I presume the story is going to become much harder, because now we have to talk about taxonomies of internal structure of organisms, of organs, and then finally of cell types. And
right.
that's perhaps one of the parts where NCBI earlier on had a role, but I don't think the community is even settled. And I'm wondering if there are plans under discussion in NCBI for how to tackle that, because the data are coming. If we don't have a good way to organize it, then the issues of storage and computing are going to be moved.
Right. Thanks for that question. I know that in the taxonomy part, we are having efforts to make taxonomy simpler as opposed to more complex. So we used to have a taxonomic ID for every strain or new strain that was described by a submitter. So for example, if this was in the past, I used to sequence an E. coli serotype X, but I happen to call this strain 12345, that would get a new taxonomic ID. Nowadays, we have stopped that practice, and we're going back to a taxonomic level that is simpler than instead of generating more and more. Now, in the case of single-cell sequencing, I am not aware of any conversations that will address this particular issue that you're raising. Now, I imagine that a single-cell sequencing becomes more accessible and cheaper, more mainstream. We will have the opportunity to think about that. But at the moment, I'm not aware of any efforts towards addressing that issue that you're raising. Yeah. Thank you.
Is there another question? Well, I have a question regarding
yes.
the storage employed nowadays for the NCBI. Maybe it could sound quite futuristic, but there has been some attempt to use the DNA molecule to decode and encode information. And well, the question is, how far do you think that the NCBI is from employing DNA as a molecule of storage game? Because we're facing this problem of storage, that we are limiting our capacity to storage such an amount of information that we are producing every day. So,
right. Okay, that's a very good question. And in fact, we are working basically in two lines. First is data compression. So, for example, SRA has the next-gen sequencing data, and that's probably the archive that is growing the most. So what we're trying to do is to develop algorithms that can compress the data very efficiently. And in terms of storage, we just buy more hardware. So if we need to store more, we just have to buy more because that's our mission. Now, we are storing this in traditional disks. So we are not exploring at the moment alternative ways of storing data like the DNA storage that you're proposing that happens more at different research universities or university setting, but we try to use technology that it has been proven to work, right? Like more commercial solutions as opposed to do research into storage. Does that answer your question?
Yes, of course.
Yeah.
Thank you. So there is another question. Dr. Leonardo. Okay, that's all.
Okay, thank you very much.